Today we will discuss another important aspect of the immune system of the human body. This system is known as the complement system. Now, as far as the immunity is concerned, you have learned a little bit about the immune system, how it protects the human body from various diseases. Now, before we delve into what complement system is, you have to know a little bit about the history of the complement system. Around about the 1800s, when the microbiological aspects were developing rapidly, scientists found out that the blood of an animal, if it is exposed to any bacteria at that point of time, was able to prevent the growth of the bacteria. Although they didn't know what is the cause and how this blood is able to inhibit the growth of the organisms, thereafter, on a series of experiments, it was found that animals like guinea pigs who had recovered from cholera, their blood, even when heat treated, was able to protect other guinea pigs from cholera. Okay. What I'm trying to stress is that they found out that blood itself could have inhibitory substances in its constituent which would prevent bacterial growth. Furthermore, when this blood was heated, it was able to protect other experimental animals in vivo. That means when it is injected into the body, it is able to prevent. However, when the same heat treated blood was used in vitro, that is in laboratory conditions, it was not able to do so. But blood samples which were not heat treated were able to prevent bacterial growth in vitro. Then, at that point of time, a scientist, Julius Bordet, he proposed that blood contains at least two types of elements. One of them is a heat insoluble and another is a heat soluble substance which can inhibit microbial agents. Okay. Thereafter, Paul Ehrlich, he actually named these two elements. The first element, which was not inactivated by heat, is what we know today as antibodies. And Paul Ehrlich gave the name complement to the other substances which were inactivated by heat, and he called it the complements. Why? Because these proteins would complement the immune activity of the or would complement the immune response against any particular bacteria. Now that was all about the history. Now as of today, I think you know our immune system has got basically two important arms. The first arm is what we call as innate immunity and the second arm is what we call as adaptive immunity. Now what basically innate immunity means? It means certain aspect of the human body protects the human body from infections which are present right from birth to death. It remains same throughout the life. Whereas the adaptive arm of the immune system changes as the human being grows, experiences newer organisms and adapts. That is the reason why it is called as adaptive immunity. Now in the innate immune system, we have two types of protective arms. The first is the blood-borne and the second one is the physical barriers. Now what is the purpose of a physical barrier? The physical barrier stops the exposure of organisms into the human being. It could be skin, it could be mucosal surface. Basically it prevents the exposure of microbes to the human body. But if a particular organism does enter the human body, then we have the innate part of the system which is present in the blood. This constitutes two parts. The first is the complement and the second are the phagocytes. Now, coming to the adaptive immunity, I think you have read some part of it. Adaptive immunity basically consists of two wings. One is the T lymphocyte or cell mediated immunity, another is B lymphocytes or humoral immunity. Humoral immunity basically produces what we know as antibodies, and cell mediated immunity actually clears all the cells, whether infected, degenerated, from the human body. Now, if we look at the whole picture, now I think you can appreciate the adaptive immunity results in the death of the human cells which are infected, defective, or damaged. The humoral immunity produces antibodies. These antibodies directly prevent entry of microorganism from blood to specific site or neutralizes the virus. Furthermore, these antibodies would also activate the complement system. I'm coming to that later. So this is the overall immune system in a gist. Again, we have to look at the immune system in two ways. The first is the primitive immune system and second is the specific immune system. Why this is important? Because the primitive immune system acts in a way which is antigen non-specific or unspecified. That means irrespective of the type of the organism, their antigenicity, this system will act. Whereas specific immune system will act based on the type of the antigens the human body faces throughout the infection. In this categorization, we can have primitive immune systems which include humoral factors. Among them, there are a lot of proteins like lysozymes, interleukins, tumor necrosis factors, cellular factors like microphages, macrophages, natural killer cells. And in the specific wing, we have cell mediated immunity from T lymphocyte that is directed, and B cells which ultimately stimulate the plasma cells to produce specific antibodies. Then let us understand where does complement stand in this immune system. I have already told you the system was named by Paul Ehrlich, and he, based on the observation, showed that these were heat unstable, that is inactivated by heat. These are a set of proteins which are present in the blood itself, predominantly secreted by the liver. And what is the role of these proteins? These proteins will inactivate any organisms, either a bacteria or a fungus or a virus, when it gets exposed into the bloodstream. Furthermore, the humoral immune system produces antibodies. These antibodies may directly neutralize a virus or a bacteria, but then it is cleared from the system by the complement system. Okay. So what would be the role of the complement system? The complement system will opsonize the bacteria. What is opsonization? Opsonization basically means that a macrophage or a phagocyte will take up a particular microorganism and will phagocyte. However, the complement system basically tags those organisms so that they are taken up at a faster rate and more effectively. Now, these complement system, I have already told you, they are a group of plasma proteins. Now, these proteins are a specific type of proteins. They are called zymogens. What are zymogens? These proteins remain in the bloodstream in an inactivated form. When a stimulus comes, these proteins will transform from the inactivated form to the activated form. And one after another, one protein will activate the successive protein, and it will what result in a cascade reaction or a chain reaction. The ultimate end goal of the chain reaction is elimination of the microorganisms. Now, the complement system acts in predominantly, it acts on three ways. Furthermore, these three ways are named as the classical pathway, the lectin pathway, and the alternative pathway. Although the first pathway is known as classical pathway, the naming has occurred because this was the first pathway that was discovered. It doesn't mean it is primitive. The most primitive pathway is the alternative and lectin pathway. Okay. You should also remember that this complement system is also present in the invertebrates. But the antibody mediated immune response is seen mainly in animals with jaws. Thus, you can understand that the classical pathway, which is activated by the antibodies, evolutionarily is a more recent phenomenon than the other pathways, although it is named as classical pathway. Okay. So, the classical pathway is activated by antigen antibody complexes. The lectin pathway is activated by lectin binding to the pathogen surfaces. I'll explain those. An alternative pathway gets activated when a particular pathogen gets exposed. All these will result in activation of the complement system. This will result in either recruitment of inflammatory and immune competent cells. First, the second result will be opsonization of the pathogens by phagocytes. And finally, this will result in direct or indirect killing of the pathogens. This is the overall outcome of the complement system. Now let us look at the proteins which are involved in this complement system. Predominantly, there are 11 types of proteins. They are named protein B, C and D. Among the C, we have 9, from 1 to 9. Now these namings are not according to the steps of involvement, but they were named as and when they were discovered. Okay. Now first part is that let us look at the classical pathway. I have already told you the classical pathway is triggered by the antigen antibody complex. And the first protein that is involved is C1. Now C1 protein is this protein. If you look closely, we have this antibody over here and an antigen over here. This is the antigen antibody complex. This is recognized by the C1 protein. Now C1 protein has 3 subunits, Q, R and S. Recognition is done by the Q, which activates the C1Q. This will in turn activate the C1R, which will activate C1S. That means the protein has 3 subunits. The Q part will recognize the antigen antibody complex, which will activate the R part, which will activate the S part. Now what will be the end result? This end result will lead to activation of a complex. First is the C3 complex. I'll explain. C3 complex, we can say, is the bridging connection of all 3 pathways. Next is we have to understand what are the convertases. Now these proteins, when they activate, they combine together to form a bigger enzyme. This activated enzyme will be the significant step which will lead to the cascade reaction, okay, or the chain reaction. This would include the convertases like C1 convertase, C3 convertase and C5 convertase. Next part is when these proteins get activated, some part go into the chain reaction, the other part breaks out. Okay. These smaller fragments will end up stimulating opsonization, and another important aspect of this part is that these proteins they are also known as anaphylotoxins. They will stimulate anaphylaxis, that is, release of histamines and other mediators in the bloodstream. What will be the outcome? Because of the release of this anaphylotoxins, there will be increased vascular permeability in the local area. This increased vascular permeability will allow the phagocytocytes to migrate from the blood circulation into the localized tissue. Furthermore, they will also trigger mast cells, which will again lead to further liberation of the histamine mediators. Now, whenever we see a complement protein, I have told you it is get activated by the previous protein, and when it gets activated, it breaks out into two forms, a smaller form and a bigger form. Conventionally, the smaller form is called as subunit A and the bigger form is called as subunit B. Well, there is an exception, that is the C2, in which the bigger fragment is CA and the smaller fragment is 2B. Recent textbooks have tried to correct it, and now they are stating that the bigger unit should be renamed as C2B. In the older textbooks, you might find that. Don't get confused. For all of the proteins, the smaller fragment is the A part, the bigger fragment is the B part. Okay. Next part would be, after the anaphylotoxins, we have what is known as formation of membrane attack complex. Now, all the activated proteins will ultimately conglomerate to form what is known as membrane attack complex. Now, the basic function of this membrane attack complex is to form what is known as a pore on the bacterial surface. Now, this pore will lead to water along with the other elements within the bacterial cell to come out of the bacteria, result in bacterial cell lysis. Other part would be the complement receptor proteins and regulatory proteins. These are also important because these complement systems predominantly act on microbial surfaces. However, they can also act on human cells itself. So, there are some of the regulator proteins which prevent irrational triggering of the complement system and also downgrading, especially after the infection has been cleared. Now, let us look at the pathways. If we look at the pathway, let us look at the first is the classical pathway. I have already told you it is triggered by the antigen antibody complex. The C1 complex will be triggered from C1q to C1r to C1s. This will result in activation of the C4, which will break down into C4a and C4b. The C4b will trigger the degeneration of C2 into C2a and C2b. Now, over here, look over here, C4b and C2a. This is known as C3 convertase. That means this will activate C3. Similarly, if we look at the alternative pathway, it gets triggered when the C3 protein undergoes hydrolysis normally. However, in presence of a pathogen in the blood circulation, the hydrolysis becomes more. This spontaneous hydrolysis will result in activation of C3b and factor BB. I have told you there are factors B, C and D. Over here the factor BB, that is the bigger fragment of the factor B. This will get activated by factors B, D and property. Now, this complex C3b and BB, this becomes the C3 convertase. Okay. You don't need to get into those details, just try to understand. In the classical pathway, the antigen antibody reaction results in C4b2a. This is the C3 convertase. In the alternative pathway, the factors B, D and properdin will activate and form C3BB, which becomes the C3 convertase. Okay. Now the C3 convertase will then activate C5. The C5 convertase, when get activated, will then, this will lead to a chain reaction in which along with the C5, C6, C7, C8 will get attached. When this is completed, multiple number of C9 proteins, say about 11 to 16, gets arranged in this circular pattern, and this complex is called as membrane attack complex. Now have you understood? These are the two main ways, the classical pathway and the alternative pathway. Now there is one more pathway which is called as the lectin pathway. Now what is this lectin pathway? This pathway in which PAMP, pathogen associated membrane proteins, they can detect some parts of the pathogen. What are these parts? Some of the sugars that are present predominantly in the bacteria, like the lectin, the fucose, the mannose, and phycolins. There are three phycolins, phycolin 1, 2 and 3. They are recognized by these PAMs, and when they attach to these carbohydrates, I have already told you the fucose and mannose, they are recognized by this complex by the MASP protein, that is mannan myelin lectin activated serine protease. MASP stands for mannan myelin protein lectin activated serine proteases. These proteases will get activated. They will trigger the formation of C3 complex, and from C3 convertase to C5 convertase, and furthermore it will lead to formation of membrane attack complex. And in all the pathways, if you look closely, the smaller fragments CA, C3A, 5A, these are liberated during this chain reaction. These will act as anaphylatoxins. Okay, have you understood? So if we look at the whole picture, what will happen? After the C3 convertase activate C3, it will convert the C5, will form membrane attack complex, and it will lead to cell lysis. All the smaller fragments will lead to release of anaphylatoxins, which will result in smooth muscle contraction, vascular permeability, and mast cell degranulation. This will ultimately result in chemotaxis, movement of neutrophils and phagocytes from blood circulation into the local site. And the C3b will result in opsonization by the phagocytes. Hence, this is the total mechanism of the complement system. Similarly, you must also try to understand there are important negative controls or regulatory controls. The first important is in the classical pathway, which is known as C1 esterase inhibitor. So it basically prevents triggering of pathway against any antigen antibody complex. Okay, because sometimes human body may even form antibody against its own cells. If it triggers on any antigen antibody reaction, it will be a vigorous reaction. Next, we have the negative control at the C3 inverse. Most important is the decay activating factor. They will compete against the bigger fragment of C4b, 2a, and will try to inhibit activation of the C3 convertase. Similarly, another important would be at the C5, in which the prolactin, it will prevent activation of the C5. These are the important regulatory proteins. Now, coming to the membrane attack complex, I have already told you C5 will activate C6, will activate C7, will activate C8, formation of a complex of C5, C6, C7, C8, which in turn will activate an insertion of C9 protein, multiple numbers, and will form a tunnel-like system. This will result in formation of a pore on the bacterial cell. Now, this C9 has a hydrophobic end and a hydrophilic end. The hydrophobic end is basically attaches with the cell membrane of the bacteria, and the hydrophilic end goes inside. Hence, water along with the other ions and electrolytes can come out of the bacteria and result in cell lysis. This is the electron microscopic picture. If you can see that on the bacterial cell wall there are multiple pores. Now, having understood that, you must understand important differences between these three pathways. The first difference is that the classical pathway is activated by antigen antibody complex. The alternative pathway is activated by, please remember, endotoxins. Endotoxins are generally produced by gram-negative bacteria. Okay, and some of the immunoglobulins like IgA, IgD, and even some toxins like cobra venom. Whereas the lectin pathway, I have already told you, it recognizes specific carbohydrate moiety on pathogens. It could be fucose, mannose, phycolins, etc. Then the first important complement that is activated in the classical pathway is C1, which recognizes the antigen antibody complex. But in the alternative pathway, it is the C3b, and in the lectin pathway, it is C4. Now, the C3 convertase in classical pathway would be C1, C4, 2a. In alternative pathway, it will be C3bb, and in the lectin pathway, it will be the mannose binding lectin and MASP C4b2a. Now, outcome of the complement function. First is the target cell lysis by membrane attack complex. The second would be inflammatory response by the mediators. Third is opsonization. Fourth is removal of immune complexes from the blood. And finally, virus neutralization. Let us look one by one. The first is target cell lysis. What happens is that the membrane attack complex forms pores and channels. This will lead to free passage of various ions and water and result in bacterial cell lysis. However, this membrane attack complex is also effective against enveloped viruses, damaged cells. It could be infected human cell or it could be tumor cells. All these can be affected by the membrane attack complex. Next comes the inflammatory response. I have already told you anaphylotoxins will lead to inflammation. Inflammation will result in increased vascular permeability, smooth muscle contraction, and chemotaxis of the neutrophils and phagocytes. Opsonization is triggered predominantly by C3b and C4b. This will tag the cells, the bacterial cells, and will ultimately be phagocytosed by phagocytes, macrophages or macrophages. Another important aspect is removal of the antigen antibody complexes. Further down the line, when you will be studying the immune system, you will study autoantibodies. Normally, human body is not affected by the autoantibodies. Why? Because if any antibody affects human cells directly, they are removed as quickly as possible. This is done by the complement system. How? The C3b is bound to the immune complexes which are recognized and attached with the RBCs. Okay. Now, these tagged RBCs will be removed by the reticular endothelial cells in liver, the bone marrow, and the spleen. This is the way the immune complexes are removed. Failure of the removal of these immune complexes may lead to many manifestations of autoimmune diseases. When these immune complexes get deposited on particular organ, those particular organs might be affected. Furthermore, when you will read about various diseases, you will understand how this can affect. The final aspect is neutralization of viruses. Now, viruses are small particles and they, I think you know that, on the cusp of being whether living or non-living. So, it is very difficult to kill the virus. What the human body does is neutralize the virus. Neutralization means this viral particle can now no longer infect any human cell. How it does that? The C3b mediates the opsonation of the viral particles. Most of the viruses will be cleared by the classical pathway. However, some viruses will be acted on by the alternative or the lectin pathway. Till now, we have understood how the complement system acts. The organisms which cause human disease, even they are evolving along with the human itself. So, some of the microorganisms have found a way to circumvent these actions of the complement system. For example, in gram-negative bacteria like E. coli and Salmonella, the long polypeptide chains, they prevent the insertion of the membrane attack complex. Another important would be Neisseria species, in which the non-covalent interaction between the cell walls prevent the insertion of the membrane attack complex. Similarly, if we look at the gram-positive organisms like Staphylococci and Streptococci, the thick peptidoglycan layer and the capsule, when present on this bacteria, will prevent the attachment of the membrane attack complex. Similarly, some of the viruses, they will start producing proteins which may mimic other proteins of the complement system. Thereby, this protein will not or will inhibit triggering of the chain reaction, like the vaccinia virus, herpes simplex virus, or Epstein-Barr virus. Okay. The other important aspect is some of the human beings may have altered or inborn errors in which some part of the proteins are missing in them. What will happen if somebody does not have some proteins that are essential for the complement system? So, if these proteins become deficient, like if C1, C2, C3 are deficient, what will happen? The classical pathway will not be triggered because classical pathway is triggered by C1. Okay. This will result in either autoimmune diseases or glomerulonephritis or pyogenic infections. Similarly, if the properdin or factor B is absent, the alternative pathway will not be activated, and the organisms which can evade the classical pathway will be causing the disease in them, like Neisseria. And finally, if some have a deficient protein which result in defective membrane attack complex, that might lead to severe pyogenic infections and disseminated Neisseria infections. Because against Neisseria, the membrane attack complex is very important. And if somebody has deficiency of the regulatory proteins, what will happen? Like the C1 esterase inhibitor, there will be unregulated triggering of the classical pathway, leading to anaphylatoxin and leading to what is known as hereditary angioneurotic edema. Hereditary means it is inherited by heredity, angioneurotic means swelling of the blood vessels, and edema because of the anaphylatoxins, that is unregulated activation of the classical pathway. Similarly, if the decay accelerating factor is there, what will happen? Complement clearing will be more RBCs will destroyed, and these destroyed RBCs will result in what is known as paroxysmal nocturnal hemoglobinuria. There will be rapid destruction of RBCs. This is a gist of some of the diseases which can occur if there is deficiency of regulatory proteins. That would be the end of the discussion regarding complement system. Now, have you understood what complement system is or not? Let us summarize what complement system is. The innate immune system has two aspects. One is the protective one, which protects the body by preventing entry of organisms into the human body. This would include skin, mucous membrane, sometimes clearing of the pathogens directly by the reticuloendothelial cells. But if it enters within the bloodstream, what will happen? The innate part of the immune system has the complement system. This system is activated by three ways: the classical pathway, alternative pathway, and the lectin pathway. The classical pathway, although named classical, evolution is a newer pathway, whereas the alternative pathway is a older pathway. Classical pathway is triggered by antigen antibody complexes. Thereby, it forms a bridge between innate immunity and the adaptive immunity. Okay. Alternative pathway is triggered by other factors of the complement like the factor B, factor D, and proprietary. And the third pathway is the lectin pathway, in which specific proteins like MASP will they will detect specific carbohydrate groups which is found predominantly in pathogens like fucose, mannose, and phycolins. After recognition, they will trigger. Whatever may be the triggering factor, most important first step is formation of what is known as C3 convertase. This enzyme will activate C3. C3 will then form what is known as C5 convertase, that is the enzyme which activates C5. Activation of the C5, that is the bigger part, C5b, will trigger a chain reaction in which C6, 7, 8 and multiple groups of C9 will bind together to form what is known as membrane attack complex. This will form pores on the bacterial cell wall, thereby leading to bacterial cell lysis. The smaller fragments of the activated proteins, predominantly the A fragments, will act as anaphylotoxins. They will trigger release of histamine mediators from muscles, increased vascular permeability, and smooth muscle contraction. Furthermore, other factors like C3b will trigger opsonization. This will trigger opsonization by the phagocytes of the pathogen. This is the pathway of the complement system. This system works along with the adaptive immune system by the antigen antibody complexes. Not only that, all antigen antibody complexes, if formed, whether autoimmune or pathogen directed, these complexes will be released from the blood system with the help of RBCs. The RBCs will get tagged with the antigen antibody complexes, and these RBCs will be removed by the reticuloendothelial cells. Now, if some of the important proteins are deficient in a particular human body, some pathogens will be, those persons will be susceptible to some particular types of pathogens. Most important, you should remember, is Neisseria species, leading to disseminated Neisseria infection. Okay. Furthermore, if some of the regulatory proteins are missing in somebody, there will be uninhibited triggering of the complement pathways. Predominant example would be deficiency of the C1 esterase inhibitor, which will lead to increased levels of anaphylotoxins, causing what is known as hereditary angioneurotic edema. And if the DAF factor is missing, then we can have what is known as paroxysmal nocturnal hemoglobin. Okay. So these are the important aspects of the complement system.